Okay, so after we've finished up, last time we were talking about the energetics of gluconeogenesis. So now we're going to talk about the substrates for this set of reactions. Okay, uh, these include things like lactate, and lactate is released by red blood cells and muscle cells. Okay, but muscle cells, it only releases lactate under anaerobic conditions. Okay, um, so during exercise. Okay, um, but red blood cells and other cells without mitochondria will release lactate as their metabolic byproduct. And that lactate can then enter the Cori cycle. All right, where it is processed in the liver back into glucose. All right. Under aerobic conditions, a muscle cell will actually be able to break glucose down all the way to carbon dioxide and water. Okay. Glycerol is another substrate for these reactions. Okay. And these are derived from the triacylglycerols. Okay. Um, these are present in the adipose tissue. Okay. They are when you get your blood taken, when you get your triglycerides measured, that's what they are measuring. They're measuring your triacylglycerol concentration, because these are produced, okay, in the fat tissue, in the adipose tissues. And so what happens is we get glycerol, okay, and glycerol kinase will consume, oops, should connect, it will consume an ATP, all right, and that will create a monophosphate of the glycerol. Right? And then we get glycerol phosphates, dehydrogenase, and that will take and produce NADH from NAD, which gives us the ability to reduce other reactants down the line, and that will give us a carboxylic phosphoester. Okay, and so this is dihydroxy acetone phosphatase. Okay, In addition, there are a couple of amino acids that can be altered uh, using pyruvate to generate alpha ketoglutarate. Okay, and those are alanine and glutamine. Right. Uh, these are the most important uh, glucogenic amino acids uh, for muscle tissues. Okay, and so they work by combining with pyruvate. Okay, and in this case, I will show. Well, I'm not going to tell you. You're going to tell me what amino acid this is. Okay. Although the enzyme might give you a hint because it is catalyzed by alanine transaminase. Okay, and that gives us another amino acid, which I expect you to identify in the comments what the product amino acid is. And it gives us our alpha ketoglutarate. And you may recognize alpha ketoglutarate from our previous discussion of metabolic reactions. Right? Eventually, this amino gets converted to urea, okay, or some other uh, alpha keto acid. That way, the amino acid balance in the liver is restored from this reaction. Okay? All right. And of course, because you've thought about the energetics of this, you know that a situation like gluconeogenesis needs to be regulated. Okay, so we stimulate gluconeogenesis with high concentrations of things like lactate or glycerol or amino acids, right? And this statement right here is just a restatement of Le Chatelier. Right? So it shouldn't be anything unfamiliar to you. Okay? These things become available, okay? They're readily available for individuals who have a high fat diet. Okay? The typical American diet, unfortunately, very high concentration of these things. Okay? Starvation. All right? When your body starts to starve, The first thing it does is it breaks down your muscle tissue to release these amino acids that it then uses to generate glucose. Okay, and prolonged fasting. Generates this as well. Okay. Allosteric regulation is summarized very, very well in figure 8.13 of your textbook. All right. Hormonally, right? Hormonally, okay, it is controlled by the insulin to glucagon ratio. Okay. If insulin is high, glucagon is low. And so gluconeogenesis will be low as a result. It's only when glucagon is much higher than insulin that we have significant gluconeogenesis. All right. So let's talk about pentose phosphate pathway. Oh, that's not illegible. Let me rewrite. All right, there we go. Much cleaner. Okay, so pentose phosphate pathway. This is an alternative glucose oxidation. Okay, so it is alternative oxidation of glucose. All right. Principal products are NADPH and ribose phosphate. Okay. It occurs in the cytoplasm. Okay. And it's 2 stages. All right. Stage 1 is 3 reactions. And it's the oxidative steps. Okay. It starts with glucose 6-phosphate being oxidized to 6-phospho D-glucono delta lactone. Okay, I want you to look up that structure and draw it in your notebook. All right. Second reaction takes 6-phospho D-glucono delta lactone that I'm not going to rewrite. Okay, I'm just going to call it product 1. Okay, so we convert product 1 to 6-phospho D-gluconate. And again, I want you to draw that in your notebook. I'm going to abbreviate it product 2. Okay. Our last oxidative or step in the oxidative chain is we take product 2 and we convert that 2, 3 keto, 6 phospho, D-gluconate. And we produce our NADPH. Okay. This is rapidly decarboxylated. To D-ribulose 5-phosphate and CO2. Okay, so the 3 keto 6 phospho D-gluconate does not last very long. We very quickly get rid of one of those carbonyl groups and eject to CO2 and are left with a D-ribulose 5-phosphate molecule. Okay. All right. The non-oxidative pathway is the other pathway, or is the other, I should say, the other uh, phase. Okay. And it consists of various enzymes. Okay. That convert the D-ribulose 5-phosphate, okay, into a variety of carbohydrate products and derivatives. Okay. I'm not going to summarize them all, but figure 8.15 on page 312 has an excellent summary. Okay. And these derivatives can then be further used to produce more energy. Okay. So this leads us to the end of the chapter, and the last section is on the metabolism of other important sugars. All right. So there are several key sugars beyond glucose in vertebrate mammals. Okay, the most notable of these are fructose, mannose, and galactose. Okay, I want you to recall that galactose plus glucose is lactose, which we get from milk, right? Mannose is a polyglucose structure, and fructose plus glucose in a particular configuration is table sugar, right? Fructose is also found in a lot of fruits, and it is found in honey. Okay? All right. Fructose metabolism, okay, has some of the most important consequences. Okay? Okay? Because it bypasses, right? It actually bypasses two allosteric regulated reactions. It jumps the metabolic pathway. Okay, in particular, it's those mediated by one of the hexakinases and PFK1. All right. So in humans, okay, chronic excess consumption right, of fructose, all right, results in greater than normal triglyceride levels, okay, in the liver, right, and lipid synthesis promoted by two enzymes, okay, SREBP1C and CHREBP. Okay. These can eventually lead to metabolic syndrome. Okay. Prediabetes is one subset of metabolic syndrome. Okay. Type 2 diabetes builds on that complication. All right. I want you to read the passage in your textbook on page 626. I want you to reflect on that. And in the comments, I want you to put in your reflection. I don't know what that was doing. All right. All right. Last bit before we hit nucleic acids is glycogen metabolism. Okay. Glycogen, you'll recall, is the polyglucose structure. It's the glucose storage molecule used by most animal tissues. Okay. Mostly made in the muscle, the liver, smaller amounts in the kidney and intestine. All right. In large part, the production of glycogen and its metabolism is controlled by insulin, glucagon, and epinephrine. Okay, so we're going to break this down. Okay, we're going to talk about glycogenesis, and then we will talk about glycogenolysis. Right, this being the making, and this glycogenolysis being the breakdown. Okay, so in glycogenesis, all right, first thing we do is we convert glucose 6 phosphate to glucose 1 phosphate, okay, which is mediated by the enzyme phosphoglucomutase. Okay, after we've got that glucose 1 phosphate, we now have to convert it to UDP glucose. Okay, so uracil diphosphoglucose, right? Okay, this you might remember from our lab on PCR as being a nucleotide, right? That makes it an excellent leaving group, right? That's the whole purpose of doing that, okay? Because this excellent leaving group allows us to overcome the endergonic nature of glycosidic bond making. Okay, so we use the hydrolysis of this nucleotide glucose bond to drive the reaction of glucose glucose bonding. All right, this reaction has overall a delta G on the order of 0. Okay, um, the uracil diphosphate will actually lose both of its phosphates, okay, to give us 2 inorganic phosphates, right? And that reaction, you'll recall, has a delta G value uh, equal to negative 33.5 kilojoules per mole. All right. So it is the breakdown of uracil diphosphate, um, which we have bound to the glucose molecule, that allows us to form the glycogen molecule. Okay. Uh, so that is our last reaction. All right. And there's 2 enzymes involved in this. All right. First, there is glycogen synthetase. All right. And glycogen synthetase, excuse me, that should be glycogen synthetase, um, adds glucose molecules to the non-reducing end of glycogen molecules. Right? There is a starter chain that's necessary, okay, which is 12 glucose bound to tyrosine 194 of the protein glycogenesis. Glycogenin. There we go. Can Dr. Cunning spell today? Apparently, yes. Okay. The other enzyme that's involved, okay, is amylo alpha 1,4,2,1,6 glucosyl transferase. Okay, and this enzyme is responsible for the alpha 1,6 branching. We observe in glycogen molecules. Okay? For glycogenolysis, there are two reactions. Okay? First reaction is removal of terminal glucoses. Okay? This is done by glycogen phosphorylase. Okay? It requires inorganic phosphate. All right? And it stops, right? It stops when it's within 4 residues of any branch point. Okay? So this enzyme chews glucose off of the end of our glycogen until it gets to a point where there's only 4 glucoses between it and a 1,6 branch point. At that point, this enzyme stops being able to function. It stops being able uh, to degrade the glycogen any further. Okay, and at this point, our glycogen is called a limit dextrin. Because we've reached the limit of this enzyme's function. Okay, that's when reaction 2 takes over, all right, which is the hydrolysis of the various alpha 1,6 glycosidic bonds. Okay, so amylo alpha 1,6 glucosidase, right, is responsible for this. And what it does is it transfers the ending 3 glucose molecules to the non-reducing end. So it transfers them to the main backbone of the glycogen, all right? And then it cleaves at the 1,6 branch point, all right? Giving us free glucose. Okay. In terms of regulation, we have our usual suspects, okay? Insulin, glucagon, epinephrine, right? Right. Um, and all the various allosteric molecules that you've learned up to this point all play key roles in the regulation pathway, right? So glucagon is released as blood glucose drops, right? Which then binds to the receptors on the liver cells, okay? right? And that generates, upregulates the generation of cyclic adenosine monophosphate, okay? Which activates glycogen phosphorylase. All right. Insulin receptors do essentially the exact opposite. Okay. It makes tyrosine kinase. It activates tyrosine kinase. All right. Which then starts triggering the phosphorylation cascade that builds up the base uh, uh, the base oligosaccharide on tyrosine 194 of glycogenin, right? So it has the exact opposite effect. It causes glucose to be stored as glycogen, okay? Epinephrine comes from the adrenal gland, okay? That and adrenaline are our two major hormones that give us our classic flight or fight response, okay? Um, epinephrine's role is to upregulate glycogenolysis and downregulate glyconeogenesis or glycogenesis. Okay. Um, in doing so, it floods our body with energy. Okay. And so our muscles are able to react faster. They have plenty of fuel ready to react, right? Which is why whenever you feel your flight or fight kicking in, um, the you feel a lot stronger. You feel like you could run a mile. You feel like you could run 2 miles if you had to, and that's because your epinephrine, your adrenal gland is releasing epinephrine, which is causing glycogenolysis. You're burning through your body's glycogen stores. Okay. Um, there's also various allosteric regulators that include adenosine monophosphate, calcium 2 plus ions that are produced by muscle contraction, glucose 6 phosphate, adenosine triphosphate. There's a lot of different regulation pathways. And I think that's where I want to leave this video. I will be posting pretty much a video every day. I think maybe tomorrow I will give you 2 videos, or maybe just one really long one. Because we're going to start talking about nucleic acids, and I definitely want to try to get us all the way through nucleic acids and into protein synthesis, because those things are very, very important. So, whatever we don't cover by Friday, though, will not be on your phone. So, I might post extra videos next week, but if it's not up by Friday, it's not on your phone. Okay? Any questions, email me, and I'll see you in the next